Welcome to STEMpaths. I'm Dr. Thornton Larson, and with us today we have our guest hosts of Preston and Dustin. Here on STEMpaths, we have students unpacking papers, study paths, and the people behind them. Our guest today is Dr. Daniel Hughes, currently at Tarleton State University. And I'll let you guys kind of take it away.
All right. The paper was... Oh, shit, I shouldn't
bleep that out. I'll do this part.
All right.
All right. Well, first of all, just thank you for being here. Like Dr. Larson said, we went through 4 or 5 papers, couldn't get any responses, so we do appreciate you being here with us. So your paper, you found that a chameleon species in Central Africa was actually 5 separate species. Just general overall gist of the paper. Am I correct with that? I know there's more to it, but trying to be broad here.
He's keeping it simple and sweet.
Title, it's that's even in the title. You can't mess that one up.
Yeah, I think that is the title.
It is the title, but I wasn't going to try and pronounce the scientific name of the chameleon. I just butcher it. So,
Rhampholeon boulengeri.
Mm-hmm.
Boulengeri.
It's a dead language, so you can pronounce it however, whatever twang you want to add to it, you can do it. No problem. There's no judgment.
Yes, sir. Was there anything you wanted to add to that summary? Because I do understand that it's more complex than just looking at chameleons.
Yeah, I mean, it's a cryptic complex, so a lot of them look very similar. So historically, people would have said it's just one species. And genetics and morphology sort of tells us a different story. And 5 seems to be the number that best satisfied the diversity there. So that's what we put out there.
So,
was there a belief that there would be more or less than what the result ended up being?
Well, that paper is a continuation of a previous paper from 2018 or 19, where we looked more deeply at the genetics. This is the morphological sort of support. So we already did have the hypothesis of 5 new species, including and then the original, so 6 total since 2018 or so. But I mean, I think in general, when you look at these wide-ranging groups and you start looking at the molecules underlying them, you don't really have a clear hypothesis about what is going to be there in terms of numbers of differences, unless somebody's described subspecies or there's very distinct coloration patterns that are different. So those are the reasons why you might have a hypothesis. But this group looks the same for the most part, as far as we know, behaves the same. So it was pure, I would say, exploratory phylogenetics.
Okay.
So essentially what he's saying is he went out there, they caught some, they did DNA analysis, and then they were able to group some of those based on locations. And some of those differences were high enough to where they would raise them to candidate species and want to then further explore this in the case of what he used morphology to kind of tease apart those relationships a little more.
Okay. Was there anything you hoped readers would glean from the article itself? Any specific pieces or notes of important information that people could take away with?
Yeah, I mean, I think if you dive into the species descriptions, one of them we named after a local term that essentially means like goddess, and we had initially named it Partheno. So the name is Nalubale, which means goddess, and then I had it initially Partheno Nalubale because Partheno means virgin, essentially, and so it was a virgin goddess because this species, nobody's ever found a male. And we've looked at, 60 or so specimens from all over the region. No males have been found. Every other species, they find males. And then genetically, they're almost indistinguishable from almost 1,000 kilometers away. They are genetically, at least for the genes we looked at, identical. And so we speculate that it is a virgin birth. These is a parthenogenetic species, which is actually somewhat common in some reptile groups, that they can do it when they're alone for a long time, like Komodo dragons. A bunch of snakes have been being found. They live in a zoo alone. Whereas in terms of an actual species that's fully parthenogenetic, there's only a couple. mostly lizards. And this has been speculated to occur in this group, but the research has sort of lagged since about the 70s. And so we named this lineage off of that sort of proposition. And so, yeah, I think that's a really cool, exciting element. And we were able to pull up papers from the late 1800s, 1900s, where they started first collecting it. And it's only females out of like 60, 70 samples.
Oh, man.
We had museum collectors go into their museums and look through them, and they only found females. So, I mean, it's not conclusive evidence, like a 100%, but if you combine the genetics, the geographic separation, lack of difference in genetics, and then the fact that only females have been found for over 100 specimens, it's pretty, for our end, strong evidence that maybe this is a parthenogenetic species. So that was cool. And then the other thing I think that stands out natural history-wise is like a lot of chameleons have been found out to do what's called bone autofluorescence, where bone, if you hit it with UV light, will automatically send off a UV signal back at you. And they have like tubercles in their face where the skin is like shortened, I would say, so that the bone is sort of revealing itself through this just a few layers of skin. And if you hit it with a UV light, it shows a really cool ornamentation to the chameleon's face. And we know that they see in the UV spectrum. And so nobody's ever documented that in this group of pygmy chameleons. And so I went through specimens and shined them with lights and got some cool images. And so one of the figures in there is that.
Well, my next question was going to be about the Nalibale, but you beat me to that one. Can you go into any more detail about how they reproduce?
Yeah, no clue.
Good answer.
Yeah, this is Central Africa. So good luck staying there long enough to actually observe some behavior. It is really a smash and grab operation in there, just really, really persistent civil unrest. And going back 100 years, even pre-colonization, it wasn't really an easy place to move around. And then once they gained independence, it got even more difficult because of militia. And so, most of the collections are done over short periods of times and 3 or 4 days here, 3 or 4 days there. We found specimens with eggs, and so we know that they're oviparous, but beyond that, we don't really know much about how it actually is achieved. And if somebody goes and looks at the genomics, maybe we could figure out if it's truly parthenogenic or not. So,
and he's touching on something that's really interesting about research in hard to reach areas. Is usually that's what it is. Right now, there's a higher focus on the phylogenetics, the phylogenomics, because it's kind of easier to do in the short term, where studies where you're going to focus on behavior, staying out in some of these remote regions, which in this case is like Lake Tanganyika, Africa, which is literally middle Africa. You're going to have challenges in spending the amount of time it would take to observe these in situ. Now, taking some back, I don't know if doing lab designs in that way would make sense for studying basic ecology for these particular species, but we'd have to learn so much about them even to get to that point.
Well, you beat me to another one of my questions, which was, what were the challenges you faced in Africa specifically?
Oh, he only touched on them.
Yeah, I mean, challenge is a broad term. I mean, it's just a difficult place to move around in. And so it depends on where you are, obviously, and there's huge variation from one country to the next. A lot of that correlates with their transition from a colonialist power to an independent state. So depending upon who colonized them, whether it was the Belgians or whether it was the British or whether it was the Germans, they left a different legacy of sort of quality and government and power, and treated the people so poorly in some cases. It was truly a coup d'etat, and there's really several coups d'etats really to arrive at some level of independence. And so, that I think just leaves a legacy of infrastructure that's usually pretty poor in some of these places, and security that's really incomplete. And that doesn't even get into the sort of current issues with like genocide that happened in Rwanda in the 90s. South Sudan right now is in a civil war. Central African Republic, right on the border. No one goes there. That's where Joseph Kony hid out, who was a notorious child soldier recruiter. That's where he would hide because it was easier to hide there than it was in Uganda, for example. So, and then that doesn't even mention mosquitoes and malaria and all of the sort of Ebola was described from the Ebola River in Congo. So there's just a whole mpox. I mean, all this stuff really is endemic there. And so just living is it can be a challenge, especially for foreigners. And so that definitely is, part and parcel of it.
We focused a lot on the negatives, but were there any positives? Did you have any rewarding aspects of your journey over into the rift?
Yeah, great question. For me, it was amazing. I think I'd speak about some of those challenges because that's what most people want to know about is how is it different than our life and blah, blah, blah. But you're in a tropical rainforest. The diversity is insane. The first time I went was with Thor and I as our advisor. And by the end of that trip, we struggled immensely with just logistics. And by the end of that trip, he told me, this was the worst trip I've ever been on. And I was like, really? This was the best trip I've ever been on. And so he had been going there for a decade. Yeah, perspective really matters. He'd been going there for a decade and had extreme success getting around. And he faced some serious troubles for sure. He's written now 2 books about his struggles. But for me as a brand new individual, just never seeing anything there before, it was all awesome. Everything was just amazing. The butterflies, the beetles, the snakes, the frogs, the people were challenging in some cases, but really curious, and some places really nice and just interesting. And so many cool stuff came out of those trips. And I've gone several times taking students, and I get to sort of see them experience that as well. So I think it's very liberating to be in a place that doesn't have access to the internet 24/7 and the ability they don't schedule meetings. We're not having this sort of meeting in Congo or Uganda. It just doesn't happen to the same degree, and there's something awesome about that.
Very opportunistic.
The island time kind of thing, only it's not island time, it's more rainforest time.
Boy.
Yeah, and it's great.
I think it's also important to point out that this study took place over several different countries in the same geographic area, correct?
Yeah.
I just want to bring that up.
Yeah, no, it's a good thing to bring up because, there's Congo, Uganda, Burundi. Rwanda's right there. What am I missing?
He helpfully included a map if you want to look at it.
Well, I know where the map's in.
Yeah, I believe too with the map. But yeah, it's a big collaboration, and samples were even collected. I think before I was born. So some of the museum specimens that we actually still had genetics were from the California Academy of Sciences, and so their samples are actually in there, and those were collected by people in Uganda in the late '80s. A guy named Robert Drews, and so we had genetic samples spanning all of those places. And then obviously we had a lot of really old specimens included as well. And so it really did take a lot of people and many years. I started this project. It was the first project that I started when I started my PhD in 2014. I started sequencing things for pygmy chameleons, and it just got published last year. So it took a decade for me, and I wasn't actively working on it every day or, there would be big lapses, obviously, but to accumulate the amount of morphological information required to sort of test those phylogenetic hypotheses meant we needed people to measure stuff in places that we weren't. And we needed the holotypes, and we needed photographs, and we needed these things. So, and that just takes time to reach out to people and sort of build up a database. So, a decade of time where somebody's actively pursuing it, that's how long it took to publish this thing. Taxonomy is the slowest field ever. It's glacial, and when you do it, people just complain often when you're done. But it takes a long time, and it really does take a lot of people nowadays, especially to get everything that's required to provide the evidence. Back in the day, you collected one new frog or one weird looking snake, new species, and you would describe it, and it's like, it could be a paragraph. I mean, Linnaeus would describe names and he wouldn't even justify why it's new. You just put a new name in there. So, it's come a long ways, which is great, but it's honestly, it's a very thankless science, I would call it. Yet it's like essential to the fabric of all other fields. You have to have a name for something if you want to study it. And if the name is wrong, it can have huge consequences.
Mm-hmm. Did you have locality issues with some of your holotypes where they were too generalized? Because I know in some of my work, I get things like the locality for a species of frog is Java.
Yeah.
The island of Java is bigger than that and has several ecoregions that are going to likely explain differences. So did you run into anything like that with your chameleons?
Yeah, definitely. And we had somebody, again, collaborator on there who knows like the original, I guess it's Germanic language that the first collector put, and there's like a use of it's in the paper, we describe it, but like I essentially had to like have him write it because I didn't understand it fully. But there's a special type of R that's used in this type of Germanic language that the original field collector in the 1900s put in there. And in one interpretation, it means like a sand mountain, but in another interpretation, it means a forested hill or something. And
boy.
Like that's a huge difference in habitat. Yeah, and it's just based on this like strange looking R. And so he sort of like, I would say not conclusively, but he definitely narrowed it down from a very general like description of like these things called sand hills to like a forested sort of mountain. And he collected some other chameleons of a different species at this one locality that he actually provided for in the same document. And so we sort of speculate that that's actually where he collected these as well. He just didn't label them correctly, which, that goes back to Darwin messing up labels. So that's always going to happen. So, yeah, I mean, I think, the standards have improved significantly because you want to get away with that today. And that generalization has a huge impact on whether it's this species or that species, right? Because the habitat is so different. But, 100 years ago, they didn't think there was probably even that many species. And they probably didn't think much about habitat-based speciation. They just thought, yeah, this chameleon looks like the other ones, got to be the same. So
I know that in captivity and domesticated chameleons, they're really hard to keep alive. Were any of the specimens you worked with kind of like that?
They didn't have to worry about that too much.
Which ones are you talking about? Because there's a huge variation. There are some you can't kill that are, released into Florida and are thriving down there, for example.
Absolutely.
But then there are others that will die if the humidity's off by a few percentage points.
Before we move on, I just want to ask, how do you get access to these places? Like I'm sure you have to talk to 1 million people to even get into this area in the first place. How do you go about that?
Well, Thor can tell you all about that. Permits and governments and interacting with indigenous scholars. You gotta have somebody boots on the ground locally, and then you have to write lots of paperwork that's very long and confusing, and then pay money and pay more money, and then hope that your permits get approved. And for me, getting over there was because of our advisor. Eli has been working there for a long time. And it's a struggle. Even he'll tell you, I mean, the amount of paperwork you have to fill out to get there, which is part of the reason why we don't take live stuff home. It's even harder because there's such a big trade, especially illegal trade in a lot of these things. All chameleons now are CITES Appendix 2. So that means they're scrutinized at airports in more extreme ways. And so to get them out alive, I mean, it's a mountain of paperwork and it's a lot more money. So if they're for scientific purposes and no monetary value and they're dead already because they're specimens, you have a little bit easier path but it's still a challenge.
And nothing's a little more. I considered my first time coming in, even though I had my fish and wildlife permits. I was always a little bit scared still that there was going to be something I filed wrong, and all of a sudden I'm going to be stopped at re-entry and have all this fun. And then working with the airlines to make sure you can travel with things correctly that are good for the specimens, as well as good for the airline to maintain their standards.
And then, go ahead.
No, you got it.
What originally got you interested in chameleons, Central African ecosystems in the first place?
Well, I kept male chameleons when I was younger, so I always loved watching them eat. I thought that was really cool. And I, again, just loved reptiles in general. And then when I was finishing my master's, I wanted to go to the equator. I wanted to go somewhere that was more diverse than the U.S. And so I just was looking for advisors that would provide that opportunity. And so when I spoke with Eli, he said, yeah, maybe you could come with me to Africa at some point. And I have a couple of shelves of chameleons that no one's ever looked at. And that was it. So I just said done, and I stopped looking at other schools, and that's where I went. And it worked out, obviously, but it was definitely a big question mark in the beginning. But that's what I wanted to do. I just wanted to go see true biodiversity, and chameleons are just something that I always liked as a kid, and it just really worked out to synchronize those things up. So
I know that the program you used for molecular analysis was the BEAST program. For phylogenetic mapping. Do you think that's the best technology for the field, or do you think that new things have come out since then that have improved?
That's a good question. I mean, I don't think there is any best. You'll see any phylogenetic paper you look at will always test their data and their trees in a couple of different platforms. Usually some sort of likelihood-based method, or with BEAST, like a Bayesian-based method, which essentially allows you to take into account molecular change and evolutionary change from A's to T's and C's to G's, and it sort of like weights that, I guess, is the best way, easy way to describe it. There's a couple more that I think have come out that handle larger data sets, like especially with genomic data sets, but they're still using the same underlying principles of BEAST or of any of the maximum likelihood-based programs. So I think the math and the algorithm doesn't change, but maybe they're a little bit more robust to uncertainty, or again, they can handle larger genomic data sets for individual species, which is going to be probably better resolution, but we're realizing the more DNA you put into a phylogenetic tree building program doesn't necessarily mean it's going to reflect accurate genealogy, because the genome of everything is a mosaic. And so this part of your genome may match you with this species, and you put those in, it's going to give you one tree. But then if you put another part of your genome, it actually matches you to another species, right? Maybe there's some sort of hybridization event that happened in ancestral time, or you have some sort of incomplete lineage sorting going on. And so depending upon which section of the genome you're looking at, you can get a couple different trees. And so I think that's going to be the big challenge for phylogenetic work going forward, is how do you reconcile the fact that we know it's a mosaic, we know that you can get speciation with gene flow. So how do you actually draw the boundary between where one species is and another is, if different parts of the genome tell you a completely different story? Which gene is more important is sort of the argument there. And people use SNPs and things like that, which I think are somewhat help that and I can address that. But then you have no clue where the SNPs come from. It's like, what? That's not, you don't even know where that came from in the genome. You just know it's a nucleotide somewhere, right? And reptiles and amphibians only have a few whole genome sequence. We're way behind. I mean, the bird people are just crushing it. Every week or every year, there's a whole new set of genomes where they're literally looking at entire genomes for a phylogeny of all bird families.
Yeah, those bird and fish people, man. Those bird and fish people.
Yeah, fish people. If we eat them or we like to look at them, we'll pay a lot of money to have research on them. And so those those have been very, very successful. And so, I think that if we could get to that level, we'll be very, I don't think a lot of the core branches in herpetology will change, like at the deeper nodes, but there's definitely going to be a lot more shuffling, especially with snakes, and some of the amphibians, like salamanders. It's so weird. We don't even really understand where they actually should be on the tree. And so I think some of that will get flipped around. But by then it's going to be machine learning. That's what they're using now to even analyze some of these genetic data sets. And that's where it's going to probably just completely outpace Beast and RaxML or IQ Tree, whatever program you want to talk about now. I think that's where it's going to really transcend.
Yeah, my experience with that is some labs are already going down those building machine learning algorithms, neural networks on top of neural networks to kind of understand that a little bit. So let me pivot a little bit, everybody, since we've been hitting some of those things. So one thing we're a little curious about is, I know you've communicated research findings in a huge bunch of different ways. What's been your favorite way to communicate research findings, both to maybe a knowing scientific audience and a more lay audience?
Yeah, I love writing. So writing papers has been great. The problem with it is, ChatGPT and things like that now, it's sort of, they're better than me. So it's like, I spend all this time, cranking out a sentence, and then ChatGPT gives me a better sentence in no time. And so, it's a little frustrating to see that, and, how quickly students will adopt that sort of technology without going through the trenches of figuring out how to write a proper sentence. But that's the thing that I really love. And I figured out that I love that during my master's. And so it really has been my favorite way of doing it. And I realize why I love it is because it allows me to read a bunch of stuff and then try to put it together in one place and try to make it make sense as a narrative that connects to my research that I did. And so I really love diving into the literature and finding cool studies and then figuring out a way to work that into the introduction or discussion or connect it to my research. And books, I love reading books. And so I think that has been really great in terms of, but you don't even know people read your papers. So, it's not like I get any sort of gratification that somebody read it. It's mostly like, I'm just glad I did it and it got through the review process and now it's out there. In terms of more instant gratification is presentations, and my last school had some cool ones where you had to come up with, or they had an opportunity where you can sort of talk about something that you think is interesting over 4 lectures to a general audience. And it was mostly geriatric folks that they'd wheel in from the old folks home, but they were really engaged and they were interested and they're lifelong learners. And yeah, they're all gray hair, but they had cool questions and they're curious about what you were talking about. And so I did 3 of those, and none of them I talked about herpetology. And I just thought it was great because I was able to take a bunch of cool stuff that I had percolating around in my head and talk to people who are actually engaged. I know Thor can say, we'll get up there and talk to students, and you don't even know if they even listen, right? Like, you'll get one student who maybe talks to you afterwards, but most of the time it's just crickets, and you're like, all right, see you next time. So you're like, do they even care? Do they even want to be there? And like, Thor and I were students longer than we've been professors, so we know that mindset. But at the same time, once you get on the other side of the desk, you do feel something when nobody says anything to you after it ends. And so to have an audience who actually would come up to you and be like, hey, that's great, or send you emails, hey, that was interesting, I'm so glad I went. You're like, oh, cool. This is like, this is awesome. So I would say that was probably my favorite.
Yeah, and on that, actually, an interesting thing is if students do come up to you and express interest in the subject matter you were talking about, and sometimes it's close enough to your research lane that all of a sudden you've got this student in your head, then you can create these undergraduate research experiences for those students because they're interested in the material. And all of a sudden their name, their personality, whatever, comes to the top of your mind when you're thinking of opportunities you want to develop.
Yeah, definitely.
Well, before we get too far, I know we're jumping around a little bit, but I just wanted to ask you, do you think you found all of the species that were previously classified as one, or you think there's going to be more in the future?
In that group, no, but more data can always tell us a different story. And so, yeah, if I wouldn't be surprised, it's hard to get surprised nowadays. So if more stuff comes out and it says that there's another population that nobody collected, and we didn't even notice, then yeah. And there was still, even in a couple of those species, there was a lot of genetic structure in a couple of those lineages. And so, one could make the argument that an isolated population is distinct enough to warrant its own name. So yeah, you could. But in terms of just like other groups in Africa, I mean, yeah, we just scratched the surface. It's 50, 75 years behind South America, and we're still describing tons of stuff from South America. So yeah, it's especially Central Africa. It's a gold mine, literally, for biodiversity and for rare earth minerals, unfortunately.
Gold and cobalt.
Yeah, sad.
Unfortunately, a lot of blood too.
I know you've got some more technical questions to ask, but I just wanted to know, I read it in the paper, it said you would go around at night with a flashlight, and the chameleons are usually lower to the ground, but how exactly did you find them? Were you just wandering around the jungle at night with a flashlight?
Yeah, that's it. You go out at night and you have a high-powered torches. Yeah, I would have a big, powerful headlamp, and then a high-powered, mag light type thing, but better than that, and then a long stick, and you just shine the trees, and you want to sort of like do it in a somewhat, speedy way, but not too fast. You just don't want to go very slow, because you're not looking at every individual leaf. You're looking for pattern recognition where something is different in the background. And what happens is when chameleons go to sleep, which they do every night, they will often walk to the end of a branch, and then they lose the ability to maintain active camouflage. And their sort of base color is like a bright green/yellow. And so when they're up in a tree that at night is completely dark, so even though the leaves are green, it looks very dark green. These guys stand out like little Christmas lights. And, once you get good at spotting them, like I did, you can find a ton. And so, yeah, very successful. I mean, we stopped collecting that chameleon in particular. In some places, yeah, I just stopped because we saw so many of them. Once I learned the technique, I was off to the races. I was finding everything. So yeah, it's fun. I really like doing that. And yeah, you use the stick sometimes because they're so high up. You gotta wake them up, and then you try to get them to crawl onto the stick, and then you bring it down. Sometimes you throw a stick and you try to hit the branch, and then they fall off and they'll roll in the air, and you just go find where they fell, and then you go pick them up. So
an exercise in reflex and catching.
Yeah, I did that with the snake one time too in Africa. I threw a stick up, hit it, snake flies off, and I snagged it in midair. I knew it was a non-venomous, this like genus called Phylothamnus, these sort of plain green snakes. It was amazing. But I was just me and my African colleagues, and they were just like, they didn't care. I was like, this is the greatest moment of my life. And they were just like,
and to them it was Tuesday.
They're like, why'd you do that? They're like, you should have just let it come down. I was like, no, what it did, look how high it is, so.
On the interpersonal aspect, your paper acknowledges Congolese and Ugandan partners that you worked with. How was the interpersonal aspect of working in those regions?
Yeah, I mean, with the people we know, the collaborators, most of which are co-authors on that paper, yeah, it's great. I mean, Eli, our past advisor, he has been working with them at that point for like a decade. Mm-hmm. So, he knew them really well. And so I was just the student that was folded in as the guy that's, going to do whatever Eli says. And so they just accepted me immediately. And, but anybody outside of that circle, I would say, especially in Congo, it's a bit challenging. Obviously I'm white and that stands out like a sore thumb. And so, you get approached by a lot of people, most of which actually don't speak English. They have 4 national languages there, mostly French, but they also have 3 others. So when they play the news, they literally do it 4 times in a row through each language, the same thing. every night. It's incredible. And so there was a huge language barrier in Uganda, where I continued to do my work and have gone back many times. That they were colonized by the British, and so they mostly speak English. And so it was more rare you'd run into a place where they don't speak English. But again, because you're white and they sort of believe you, and if you show up and say, I'm here to catch snakes and chameleons, but they also are suspicious because they've been screwed over by people that look like me for a long time. And so there was moments for sure where I got cheated out of stuff, or, they hiked up the prices significantly. They did something to sort of subvert sort of our goal. And mostly they did that through speaking local languages that I didn't understand. But if you have a trusted collaborator and a colleague that's from there, they will say in the local language, oh, no, don't do that to this one. He's one of the good ones. And let's not cheat this guy. Or he'll come to you and say, hey, they're trying to cheat us. Let's give them a little bit more money. So we won't pay, this much. We'll pay like this much because I've talked to him, So, yeah, but in some places we were definitely welcomed warmly, and the reception was great. But in a lot of places, yeah, they do want money. They're struggling in a completely different way than we could ever even imagine in most of the U.S. And so that it totally made sense. And it didn't really bother me, to be honest, after a little while. Once I realized that I was being cheated, I thought, who cares? I'm still getting what I need out of these situations. And yeah, and I mean, I still work with them, I still collaborate with them, and they're happy to continue to do so.
If I recall correctly from conversations we had years ago, wasn't there some superstition about you specifically, given your distinct hair color and vampirism, as I recall?
Well, in a place in Congo, Eli had a drink mix, like a Crystal Light, but it was red. And he would pour it into his water bottle, which, he'd remove the label. And so he had this big red bottle, and he would be drinking that. And in this place in Congo, it's actually known, it's been written about, that they believe that white people are vampires. And so we got stuck there, our truck did. And yeah, Eli's drinking this big red bottle, and people were like running away, and it was really weird. And we were like, didn't understand it. And then when we started driving again, this like lady emerged out of the forest and saw Eli and myself in the car and just like took off running. And we were like, oh no, we thought she stole something off the car because everything was on the top. And we just saw her run after she saw us. So we thought, oh no. So we sent someone to chase her down. And they eventually just found out she saw us and thought we were vampires there to drink her blood. And, just got scared and ran away. And they came back and told us that. And I just looked at them thinking, you're making this up. This is a lie. And no, sure enough, it's like a local sort of legend that if you have white skin, you're a vampire. And like, yeah, and Eli had that big Kool-Aid. Red bottle. Yeah, he had the big Kool-Aid bottle. It was like
that doesn't help. No.
I was like, you did not help this situation. He was like, I did not know this.
Stick to the blue flavors.
Yeah. The blue flavor.
Then they'll have more questions.
Well, Dr. Hughes, let us switch over to the wrap-up questions here, since we're about at time, so we can finish you up. We just got 3 questions we ask every person who comes onto our show here, and so I'll let them ask those last 3 wrap-up questions.
Yeah. What's one piece of advice you would give your undergraduate self from the past?
In terms of what I do now, or in terms of just like, maybe don't drink that extra, for that one night?
Just something that you would tell yourself from the past to make sure you would do better than whatever you did in the past.
I think for me, I didn't. Okay, sure. Yeah, in terms of where I am now, I didn't really know that this was a possibility. So I think if I started that pathway a little bit earlier, I'd be in a stronger position, or at least more aware of sort of what opportunities exist. And I didn't really start thinking about it till almost the end of my junior year, approximately. So that's pretty late in the ball game now that I know. And so I would say start earlier. Start thinking about life after undergrad a little bit sooner.
Right.
Do you have any other interests if you weren't doing this? Is there anything else? Or is this your...
If I wasn't doing herpetology,
Yes, sir.
If you weren't in the
Yeah, I was gonna
researcher
go-
world.
yeah, I was going to go into nutrition. That was my other interest. So it'd be something in that world. I don't even know if it would be research, to be honest.
What would you like the general populace to understand about science and scientists as a whole?
Yeah, I think maybe that it's a process, and then it's subject to revision, and it's also a big part of a product of our culture, like meaning that you are a human making inferences about data in the world, and you bring your biases to it, and so it's not like we're seeking some sort of ultimate truth that exists in the world. We're sort of trying to explain what we see in front of us through hopefully a rational lens and hopefully with objective measures. But I think sometimes outside says, well, scientists are pure and they don't make mistakes and they go for only the objective truth, when in reality, it's just not the case. And science is really good at actually revising and changing and updating, that they don't stick with ideology, except usually within that generation. It's like this is the current ideology. We all go with that, and the next generation goes, "Wait, hey, they were wrong for these reasons, or they were right for these reasons. Let's revise the system and move forward." And I think that gets lost in the sauce of communication outside of the school. That we're people, we make mistakes. It's definitely trying to be better in some ways, but just like every profession, it's done by humans, and it's not perfect, and it's not necessarily the ultimate pure truth that's out there. We're just trying to infer with our abilities. Looking back in 100 years at what we've done now, we're going to be like, those guys were crazy. Why would they think this? Yet we're living in that lifetime thinking this is exactly the way it always is. Those guys 100 years ago were wrong. Look how stupid they were, right? So, yeah, I think that gets lost for sure.
Mm-hmm.
With that, I want to thank Dr. Daniel Hughes for making some time to come talk to us today. I want to thank Dustin and Preston. I almost forgot your name this time. For coming and hanging out with us today. This has been STEMpaths. I'm Dr. Thornton Larson, and we'll see you next time. Thank you.
Thank you.
Yes, sir. Thank you.